Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with Shatreen Sawadi. And today I am here with ribonucleic acid. Yes, this is the second most important type of the nucleic acid. In my previous lecture, I have discussed in detail about the deoxyribonucleic acid. And today I am going to share the ribonucleic acid, which is abbreviated as RNA. And in this lecture, I will discuss about its discovery and where is RNA made, structure of RNA, types of RNA, its occurrence and role in protein synthesis. Okay, first of all, I am starting with the discovery of the RNA. Its discovery is so long, but I am just here sharing a little bit about its discovery. The discovery of RNA began with the discovery of the nucleic acid by Friedrich Mischer. I have discussed about the Friedrich Mischer with his photograph in my previous lecture about the DNA, so must go through that lecture. In 1868, who called the genetic material as the nuclein, and since it was found in the nucleus. The concept of messenger RNA emerged during the late 1950s and is associated with the Crick's, and you are familiar with the name of Crick, Watson and Crick, very familiar for the DNA double helical structure. So Crick's description of his central dogma of molecular biology. And what is central dogma? You know what is central dogma? Yes, central dogma means the transcription and translation. The first DNA formed the RNA, messenger RNA, and then that messenger RNA in turn led to the synthesis of the proteins. Is it clear? Okay. This is a little bit of discovery. Where is RNA made? Many students ask me about the synthesis of the RNA, where RNA is made. They are confused, so I'm here solve your issue and answer your question that in case of the prokaryotic cell, you know that the prokaryotic cell lack the nucleus and the eukaryotic cell has nucleus. So in case of the prokaryotic cell, DNA which is present directly in the cytoplasm, you can see here DNA is present, and that region is known nucleoid. Is it clear? So DNA form the RNA, and all types of RNA like messenger RNA, transfer RNA, ribosomal RNA, all synthesized in this prokaryotic cell. But in case of the eukaryotic cell, what will happen? All the three types of the nucleic acid, ribonucleic acids, messenger RNA, transfer RNA, ribosomal, synthesized within the nucleus. Is it clear? So transcription occur here and translation occur here, and all the types of other types like transfer RNA and ribosome are also synthesized here. So what will happen? In case of the prokaryotic cell, types of RNA synthesized in the cytoplasm, and in case of the eukaryotic cell, synthesized in the what? In the nucleus. Is it clear now? Now you are not confused you will be not confused about the synthesis of the RNA in case of the prokaryotic cell and in the eukaryotic cell. Okay, now I'm going to share about the structure of the RNA. What is the structure? You can see here the helical structure. This is just single helix, single strand. Like in case of the DNA, this is the double stranded or double helical structure. But this is just single. Okay. Now, structure speaking, ribonucleic acid is quite similar to DNA. However, whereas DNA molecule, both must keep it in your mind, DNA and RNA, they are type of what? Polynucleotides. There are different types of nucleotides, like I will discuss in my second lecture, inshallah. Mononucleotides, dinucleotide, polynucleotide. So RNA and DNA are the type of what? Polynucleotide. So both are polynucleotide, but the ribonucleic acid is quite similar to DNA. However, whereas DNA molecules are typically long and double-stranded, but the RNA is what? They are much shorter and typically single-stranded. RNA molecules perform a variety of roles in the cell, but are mainly involved in the process of protein synthesis, means the central dogma, transcription and translation, and its regulation. So RNA are most familiar for the transcription and translation, mean the protein synthesis in the cell. Is it clear? Okay. Now, the RNA, which is single-stranded and is made up of ribonucleotide, like DNA is made up of deoxyribonucleotides, must clear your concept that the deoxynucleotides Like there are two types of nucleotide here in case of the DNA and RNA, like ribonucleotides and deoxyribonucleotides. Deoxyribonucleotides are present in the, and why they are called deoxy and deoxyribonucleotide and ribonucleotide? The reason I have discussed in my previous lecture. So must watch that lecture, why it is called the deoxy and why it is called the oxy. So ribonucleotide in the RNA chain contain same ribose, three groups: ribose, 4 nitrogenous bases and phosphate. But in case of the nitrogenous bases, adenine, uracil, guanine and cytosine, because here uracil is present instead of the thymine. Is it clear? In case of the DNA, what will happen? The uracil is absent and thymine is present in case of DNA. But in case of the RNA, instead of the thymine, uracil nitrogenous base is present. Is it clear? Okay, you can see here the nucleotide composition: ribose, 4 nitrogenous bases and phosphate group. And these ribonucleotides and nucleotides are linked by the phosphodiester bond, and I have discussed in my previous lecture about the phosphodiester bond and the complementarity, each and everything, so must go through that lecture. Okay. Now you can see here the difference or comparison between the this is double helical structure, this is the single helix, you can single strand, and you can see here the nitrogenous bases too, adenine, guanine, cytosine, thymine in case of the DNA, and in case of the RNA you can see here adenine, guanine, cytosine and uracil present. Is it clear? So this is the secondary structure, RNA present in that form. Is it clear? Coiled around each other. And sometime in case of the RNA, what will happen? The both strands coiled around each other and form the hydrogen bonding with its own strand. Is it clear? This is the secondary structure of the RNA. Okay. Now, I'm going to share the types of the RNA. What are the different types of RNA? You can see here the very beautiful figure of the RNA with its nitrogenous bases, purine and pyrimidine, purine, adenine and guanine, but pyrimidine, cytosine and uracil, the secondary structure of the RNA as well as the nucleotide of the RNA. You can see here, this is the ribonucleotide, which consists of the ribose sugar, phosphate group, ribose sugar and what? Nitrogenous base. So there are 3 types of the RNA which I am going to share. There are many more types of the RNA, but here I am just sharing the 3 types, the most important types of the RNA. These are messenger RNA, transfer RNA and ribosomal RNA. Many non-coding RNA I will discuss inshallah later on. Okay. Now, first of all, I'm going to discuss the messenger RNA. What is basically messenger? Messenger RNA is relatively unstable and short-lived in the cell. Must keep it in your mind. This is unstable, and unstable means it is short-lived, especially in prokaryotic cell, ensuring that proteins are only made when needed. When proteins needed in the cell, needed by the cell, what will happen? The messenger RNA is synthesized and perform the function of transcription and translation. Otherwise, its formation stop in the cell. The number of nucleotide will be more than 12 and 24, but less than 48. So it is short and unstable. Messenger RNA contain codons, and each codon has 3 nucleotides. I have discussed in detail about the codon genetic codes in my lecture about the codon degeneracy, so must watch that lecture. Messenger RNA contain many codons. Like you can see here, there are 12 nucleotides. So 12 mean there are how many codon? Because each codon consists of 3, there are 4 codons. If we are saying that there are 12 nucleotides, then there are how many codons? There are 4 codons. So each codon, you can see here 1, 2, 3. This is messenger RNA structure which is synthesized from the DNA. And you can see here 1, 2, 3. This make the one codon, then this is the second codon, this is the third codon, this is the fourth codon, and so on. Is it clear? And each codon genetic code responsible for one amino acid. I have discussed about the codon degeneracy too, that what is codon degeneracy. I am here not discussing again the codon and codon degeneracy, so must watch that lecture. Okay. Now, the function of messenger RNA. As we are familiar that messenger RNA play a very important role in protein synthesis. As you can see here, this is the messenger RNA which attached to the ribosome in the cytoplasm, and showing this diagram showing the translation in the cytoplasm. What? Translation. In translation, what will happen? Messenger RNA from the nucleus come into the cytoplasm and attach with the ribosomes. Is it clear? So the other types of the ribosome like transferRNA come and translate the message of the messenger RNA, and then bring the amino acids from the cytoplasm and make the polypeptide chain. Is it clear? So this is the messenger RNA carry the message from the DNA which control all of the cellular activities in a cell. If a cell requires a certain protein to be synthesized, the gene for this product is turned on. Is it clear? It turned on means it become active and produces the messenger RNA. So messenger RNA is synthesized through the process of transcription in the nucleus. And then what will happen after transcription? The messenger RNA move toward the cytoplasm and then attach with the ribosome, and then transfer RNA come and translate the message of the messenger RNA, and then pick up the amino acids from the cytoplasm and bring it to the ribosomes and make the polypeptide chain. That is the translation. Is it clear? Okay. Now transfer RNA. Transfer RNA is the third main type of the RNA and one of the smallest, usually 70 to 90 nucleotide long. This is about 70 to 90. This is much longer than messenger RNA. Messenger RNA is just about 12 to 24. Is it clear? But this is about 70 to 90 nucleotide, and you can see here the structure of this is transfer RNA. And transfer RNA, you can see here, this is the site where this is the amino acid attachment site. Here amino acid attach, and you can see this site. This is the portion which contain the anticodon. Anticodon also consists of 3 nitrogenous bases. Is it clear? I have discussed each and everything related to codon and anticodon, so must watch my lecture about the codon and anticodon. So here just you can see, and this portion which contain the anticodon, read the codon and pick the amino acid towards the what? Towards the ribosome for protein synthesis. Is it clear? So the basic function of the transfer RNA is to pick up the amino acids after reading the message of messenger RNA. So it carries the correct amino acids to the site of protein synthesis. As you can see here, the transfer RNA pick up the amino acids and then bring it to the ribosome. It is the base pairing between the transfer RNA messenger RNA that allow for the correct amino acid to be inserted in the polypeptide chain being synthesized. You can see here 1, 2, these are the transfer RNA which come and first read the message of the messenger RNA. You can see that this is the codon, this is the anticodon. Is it clear? So what will happen after reading the message of the codon, and then transfer RNA pick the amino acids from the cytoplasm and then back to the ribosome for the formation of the polypeptide chain, which is known as translation. I am repeating again and again, so I hope the lecture will be, the point will be clear upon you people. Okay. Now ribosomal RNA. Ribosomes are composed of what? Ribosomes are composed of ribosomal RNA and protein. As the name indicated, they're ribosomes. You know that ribosomes, which are the small granule-like structure, which are responsible for the protein synthesis. These ribosomes are synthesized in the nucleolus of the cell, means the eukaryotic cell. But in the case of the prokaryotic cell, there is no nucleolus. So ribosomes synthesize directly in the cytoplasm. But in the eukaryote, what will happen? The ribosomes are synthesized in the machinery, in the machine that machine is known as nucleolus. Nucleolus, which is present in the nucleus of the cell. Dense organelle, small, very suborganelle nucleolus. So ribosomes, which are made from, are composed of ribosomal RNA and protein. As its name suggests, ribosomal RNA is a major constituent of the ribosome. Ribosomal RNA is what? This is the major constituents of the ribosomes, composing up to about 60% of the ribosome by mass. So a ribosome is made up of about 60% by mass of what? Ribosomal RNA. And ribosomal RNA also synthesized from the DNA, and that ribosomal RNA help in the formation of ribosome. Ribosomal RNA providing the location where the messenger RNA binds. Is it clear? So you can see here, this is the ribosome, and this ribosome consists of the two units, largest unit and smallest unit, and these units are made up of what? Ribosomal RNA and protein. So the function is here. The number one, must keep in your mind that the function is ribosome formation and also providing the location where the messenger RNA binds to the or attached to the, you can see here, this is the ribosome, and messenger RNA come and attach with the ribosome here. Is it clear? So it providing the site for what? Messenger RNA binding. Is it clear now? Okay, very interesting and very easy. In bacteria, the small and the large ribosomal contain about 1,500 to 3,000 nucleotides respectively. This is much longer than the messenger RNA and transfer RNA. How many nucleotides? 1,500 to 3,000. Is it clear? In bacteria. Whereas in human, they have about 1,800 to 5,000 nucleotides respectively. So this is the longest RNA type. Okay, now function of ribosomal RNA, as I have discussed that the function of ribosomal RNA is in the formation of what? Ribosomes, and it also provides the site for the attachment of the messenger RNA. So the ribosomal RNA ensure the proper alignment of the messenger RNA, transfer RNA and the ribosome. The ribosomal RNA of the ribosome also has an enzymatic activity. Must keep it in your mind. It has enzymatic activity, and that enzymatic activity is related to peptidal transferases. This enzymatic activity is referred as peptidal transferases and catalyze the formation of the peptide bonds. You know that the amino acids are attached with each other with the help of the peptide bond, like the nucleotides are attached with the help of the phosphodiester bond. Is it clear? So the peptide bond is facilitated by or formed by peptidal transferases and catalyze the formation of the peptide bonds between two aligned amino acids during protein synthesis. Very interesting. Very interesting. So for your information, I have something. Ribosomal RNA and transfer RNA are stable types of the RNA. But messenger RNA is what? I have discussed that this is unstable, short-lived. In prokaryotes and eukaryote, transfer RNA and ribosomal RNA are encoded in the DNA, then copied into the long RNA molecule that are cut to release smaller fragments containing the individual mature RNA species. Is it clear? In eukaryote, synthesis, cutting, assembly of ribosomal RNA into the ribosome takes place in the nucleolus of the nucleus. Is it clear? In prokaryotes, these activities occur in the cytoplasm. I hope so. You enjoyed this lecture, and if you have any question yet, write in the comment box. I will try my best to answer your question. Inshallah, in my next lecture, I will I will explain nucleotides and its types. Thank you. Till then, Allah Hafiz.